Hello friends, myself Professor Khemchand Surana. So in last session we have started the new chapter, that is what anthelmintic drugs or antihelminth agents. In last session we have seen the introduction of these helminthiasis or the antihelmintic agents. In that we have seen again further the different types of these parasitic worms, that is what the roundworm, flukes and tapeworms basically. Then further we have seen this pictorial diagram and the basics of these anti-helminth or anthelmintic drugs. In that we have seen the different terms, that is what the vermicides and vermifuges. So further we have discussed about the anthelmintic drugs classification. So till that we have seen in the last session. So today we will start the classification and details of every drug or the class from the anthelmintic agent. So the first major class from this anthelmintic agent is benzimidazole derivatives. This is what the important class as per your examination. So on this class there will be the question on the final examination, so need to remember. So the benzimidazole derivatives as an anthelmintic agent. So first class, so these are the versatile anthelmintic agents particularly effective against the gastrointestinal nematodes. So already we have seen in last session, so there are different kinds of parasitic worms. So specifically we were called it as a metazoa, and these were again further divided into three different types, that is what the nematodes, trematodes and cystodes. So these are the what benzimidazole agents were effective in against the gastrointestinal nematodes. So these class were basically and particularly effective against the gastrointestinal nematodes. These are highly effective against Ascaris, Enterobius, Trichurias and hookworms infection as a single or mixed infections. So if we'll see as the point of view pharmacology, so there are what different species of these worm parasites. So we didn't gone through in detail of these different species, but as per the basic mode of action of these drugs, so whatever these benzimidazoles are highly effective against either Ascaris, Enterobius, Trichurias, hookworms infection as a single or mixed infection. So these agents were effective against these different kinds of species of the worms. So the benzimidazole derivative, so the basic structure, so as per the medicinal chemistry point of view, we have to go through the structures of these benzimidazole. So the different and simple structure of these benzimidazoles having, so the basic structure is given over here. So you can see, so benzimidazole ring is there, NHCOO CH3 is there, and R is what substitution with different drugs. So as per the benzimidazole different examples, the first and most important drug is albendazole. So R group we have to substitute. What you have to do to, so write the basic structure in your notebook basically, and replace the R from every example and draw the every structure of this class. So the first is what albendazole is there. So albendazole at the position of R is CH2, CH2, CH3 is there. Then second major example is mebendazole. So COC6H5 is there at the position of R into the basic structure. Then flubendazole, C double bond O, benzene ring and fluorine is there. Cyclobendazole, there is a cyclopropyl ring, C double bond O. Then tribendazole, then cyclohexane SCH2 is there. Then fenbendazole SC6H5, oxybendazole OCH2CH2CH3, perbendazole CH2CH2CH2CH3. So these are what the different examples of the benzimidazole derivatives as an anthelmintic agent. So mebendazole, albendazole, then flubendazole, cyclobendazole. As per exam point of view, you need to at least remember the four structures from this class. So these are what the different structures of benzimidazole derivative. You just draw in your notebook. So the mode of action of this benzimidazole derivative. So what is this mode of action of this benzimidazole derivative is, these drugs act by blocking the glucose transportation. So how they will act, basically blocking the glucose transportation in parasites and leads to depletion of glycogen storage of the intracellular microtubules in the cells of worms, thereby arresting the nematodes and cell division in metaphase. So the simple mechanism is there. So what they will do, so they will block the glucose transportation into the parasites, and because of blocking these glucose transportation, there is depletion of glycogen storage into the intracellular microtubules of the worms. Because of depletion of these glycogen storage, there is what the arresting the nematode cell division into the metaphase. So whatever the we have seen the basic cycle of cell division, in that particular metaphase is there. So by depletion of these glycogen storage, the whatever these cell division phase will be arrested into the nematodes. So the major site of action is microtubular protein beta tubulin of the parasite. So the mode of action is quite simple, blocking the glucose transportation, which leads to glycogen storage and resulting in arrest the nematode cell division into the metaphase. And the what is the major site of action is there, the microtubular protein beta tubulin is what the site of action of this parasite. So these drugs are bound with beta tubulin and inhibit the polymerization. So whatever these drugs are doing, they were bind to the beta tubulin protein and inhibit the polymerization. If the polymerization is inhibited, there is what the whatever the concentration of these parasites where decreases. So this is what the simple mode of action of benzimidazole derivative. So further the metabolism of these derivative is, so the first is given over here, the metabolism of albendazole. So the parasite, whatever the parent compound is rapidly and almost completely metabolized by oxidative and hydrolytic processes. So how they were metabolized, basically? So they were completely metabolized by oxidative and hydrolytic processes. So oxidation and hydrolysis, these are what the two processes of metabolism. So the phase 1 oxidation reaction is commonly carried out. So initially what the phase 1 oxidation reaction is carried out by using the cytochrome P440 enzyme, which may followed by the phase 2 conjugation. So initially what happened, the phase 1 oxidation will be carried out by using cytochrome P440 enzyme, and further after oxidation, so phase 2 conjugation will be occur. So this is what the metabolic processes of these whatever the benzimidazole derivatives. Again further into this whatever the reaction given the metabolism of albendazole is there, basically. So initially what they were metabolized into the albendazole sulfoxide, you can see the structure. So albendazole, then whatever that R1 will be the particular substitution, and S sulfur is there, then O minus C3H7. This is what the active metabolite of this albendazole. Further it will again metabolize into again another metabolite. So this is what the basic metabolism of albendazole. So further the here given the metabolism of mebendazole, the drug from same class. So mebendazole is metabolized by the reduction of 5-carbonyl group to secondary alcohol. So initially what happened in case of mebendazole, so reduction of 5-carbonyl group. At fifth position there is what carbonyl group. So you can see the structure of mebendazole. Initially what the benzimidazole, the first number from the NH, then second amino group, third again nitrogen, fourth from this benzene ring, and at fifth position there is what carbonyl group. So in case of, you can see the structure, there is what the carbonyl group at fifth position. So what happens, the reduction of carbonyl group of fifth position into the secondary alcohol, which greatly increases the water solubility of this compound and thereby potentiate the excretion through urine. So what happened basically, so initially whatever the carbonyl group at fifth position, it will be metabolized into the secondary alcohol, and because of this conversion of carbonyl group into the alcohol, it will increases the water solubility of the structure, and because of water solubility it potentiates the excretion through urine. So again, secondary alcohol and amine readily conjugated. So whatever the further secondary alcohol metabolite will be there or amine metabolite will be there, they were conjugated. So into this reaction or the pictorial diagram, you can see mebendazole initially given again by arrow. So amino metabolite is there at R1 position, you can see, means at second position there is NH2, means amino derivative or amine derivative of this mebendazole. So it will again they subjected to conjugation and their conjugates were formed. Similarly at second reaction you can see mebendazole. So there is what OH given over here, why OH? Initially what it has reduced through this reduction metabolism pathway, the OH over there. So this is what the hydroxy metabolite of this mebendazole. Further it leads to conjugation and forms the conjugates. So this is what the metabolism of mebendazole drug. Then further the metabolism of thiabendazole is there. So drug from similar class. Thiabendazole is metabolized through aromatic hydroxylation at fifth position and catalyzed by CYP1A2. So this is what the initially they were leads to what aromatic hydroxylation at fifth position by using CYP1A2. So this is what the aromatic hydroxylation subjected. Further, the whatever the phenol conjugates, whatever the resulting phenol is conjugated through to the 5-hydroxythiabendazole glucuronate. So you can see the reaction thiabendazole is there. So initially what it leads to aromatic hydroxylation. Aromatic hydroxylation means what? At aromatic ring there is what hydroxyl group substituted. So in second structure you can see OH over there. Further it leads to glucuronic acid conjugation and it forms their glucuronic conjugates metabolites or glucuronic conjugates, or you can call the glucuronic metabolites. So this is what the metabolism of thiabendazole. So next is what the synthesis of albendazole. So which is what the important as per your examination point of view. So synthesis of albendazole will ask for 3 marks in the final examination. So synthesis is given over here, you can see. You just need to draw these synthesis. So the starting material is 3-mercaptophenyl acetamide, benzene ring is there, SH, NHCO, CH3. In the presence of these alkyl bromide, CH3, CH2, CH2Br, there is what S alkylation will be there, means at sulfur there is what alkylation will be happen. So you can see the hydrogen from this SH will replaces by this alkyl group CH2, CH2, CH3. You can see the first intermediate. By further, in the presence of HNO3, the nitration of this will occur. So at second position there is what NO2 will be there. So this is what again second intermediate. Again in the presence of NaOH and H2PD. NaOH is for what? Hydrolysis of this amide. So in this intermediate NHCO CH3 is there. By using NaOH, hydrolysis will be there. So NHCO CH3 convert into NH2, and at second catalyst, that is what the H2 and palladium, hydrogen and palladium. So whatever NO2 will be there, that will reduces into amine. Further, in the presence of these thiocyanate ester kind of the starting material, so this starting material is not conventionally available. We just had to prepare initially into the laboratory, then we can use. So by using this we can get the albendazole, means what? There is what cyclization between these starting material and the intermediate, so which leads what? Albendazole synthesis. So this is what the synthesis of albendazole important as per your examination point of view. So the second major category is quinoline and isoquinoline. So this category we'll see into the next lecture. Till that, happy learning and